Hi everybody, Dr. Ahles here. In this short video, we're going to talk about the first two special senses. The first one being olfaction, the sense of smell, and the second one being gustation, the sense of taste. Let's start with olfaction, the sense of smell. When we talk about olfaction is technically defined as detecting chemicals in the environment. These chemicals are specifically called odorants. So odorants are the chemicals that you detect when you smell things. Because we're detecting chemicals, I want to draw your attention to the top corner of this slide. Notice that olfaction is something that we use chemoreceptors to do. So we're tying in those types of receptors, chemoreceptors, the ones involved in the process of olfaction. So when we smell something, we are smelling an odorant, a chemical from the environment. For us to be able to smell it, it must be dissolved in mucus. Once you've written the word mucus, I want you to underline, highlight, star it. For you to be able to detect an odorant, it must first dissolve in the mucus that's in your nose. This is the primary job of that mucus, to dissolve these odorants. If we didn't have enough mucus, if our nose was very dry, that would impair our sense of smell, nothing to dissolve those odorants in. And the opposite is true as well. If we have too much mucus, we dissolve the odorants, but they get completely diluted and we don't smell them either. So we need to have mucus to dissolve the odorants, but not too much and not too little to make sure we can smell them. The olfactory epithelium is where we do smelling, and there's this small little 1 inch by 1 inch square inside your nasal cavity. And if we zoom in on that olfactory epithelium, we see several different types of cells found inside of it. So the first kind of cells are the olfactory neurons, and I want you to underline, highlight, star that these are bipolar cells. Bipolar. Remember that when we were learning our classifications of neurons, the only place we had bipolar neurons was in the special senses. When you have a bipolar neuron, we have dendrites that all fuse together, go to the cell body, and it extends off of the cell body in a single axon. So we have one group of things going into the cell body, one group of things leaving the cell body. This is why we call these bipolar neurons. So my olfactory receptor cells, my bipolar neurons, are covered in a special kind of receptor called a metabotropic receptor. I don't need you to go through and learn all the specifics that are involved in a metabotropic receptor, but here's what I do want you to write down by metabotropic. I want you to write down that this is a series of events. Series of events, or multiple events. We don't directly activate ion receptors when we talk about a metabotropic receptor. So here's how it works. I have an odorant in my environment that attaches to my olfactory receptor, but the olfactory receptor doesn't have an ion channel attached to it. When that odorant attaches to my receptor, it's going to activate some proteins inside, which are going to activate some other proteins, which ultimately are going to activate my channel protein that allows ions to rush inside, leading to me detecting an odorant. So when you see this word metabotropic, just know that it's multiple events in a line. It's a series of events for me to be able to get a charge developed. In addition to having our bipolar cells that are covered in metabotropic receptors, we also have supporting cells in the olfactory epithelium. Those supporting cells function a lot like the neuroglia in the rest of our nervous tissue, so providing structure, providing protection and nourishment. All of those are functions of the supporting cells. We also see basal cells in the olfactory epithelium, and the basal cells are a special kind of cell that are able to differentiate or reproduce into new bipolar neurons. So what this means is about every 2 months, you completely trade out all of your olfactory receptor cells and make new ones with these basal cells. So basal cells make new hair cells, or excuse me, we're calling them bipolar. I apologize. Bipolar neurons. My supporting cells help those bipolar neurons to function, and the bipolar neurons are what actually detect the odorants. When we talk about smelling, olfaction is a sense that we would say has a very low threshold. We talked about threshold when we talked about pain, so here's a quick reminder that threshold is the level that a stimulus has to be at for you to be able to detect it. So when we say olfaction has a low threshold, what we're saying is you don't need very many odorants to be able to smell something. So the first thing to know about smell, it doesn't take a lot of chemicals for you to detect it. The second thing for you to know about smell is that this is a rapidly adapting sense. Remember when we talked about adaptation, it means that you initially detect something, but you may not keep detecting it going forward. You can see just how fast olfaction adapts when we start to look at the timing. So after about 1 second of smelling something, you rapidly stop smelling it. You can still smell it to about 1 minute, but once we hit the 1 minute mark, you're not going to keep smelling that sensation anymore. So olfaction rapidly adapts, meaning you're not going to keep smelling things forever. You'll smell them initially, but your brain will very quickly filter them out. And when we talk about smells, there are 2 kinds of processing that we do with smell information. There's both conscious processing and unconscious processing. So the conscious processing occurs in the area of the brain called the olfactory cortex. This was that small section on the temporal lobe that we always had an arrow pointing to saying this is where smell goes. So the olfactory cortex in the temporal lobe, this is where you consciously process what you're smelling. But there's also a lot of unconscious processing that happens alongside the conscious processing. So smell information comes in, and initially it's going to go into the hypothalamus. And the hypothalamus helps to sort it to other parts of the brain as well. Places like the hippocampus, where we store memories about what we've smelled in the past. Places like the amygdala, where we might have an emotional response to the smell. Places like the reticular activating system that helps me know if I need to have some kind of visceral response, my organs respond. All of these happen at the same time as you're consciously perceiving a smell, or even before we consciously perceive a smell. So the process of olfaction occurs in the olfactory epithelium, that small 1-inch square in the nasal cavity. We process that information in the temporal lobe and in several other parts of the brain. It doesn't take much information for us to be able to smell something, and also we adapt to it really fast. That is all we need to know about olfaction. Let's move into our next sense. Our next special sense is gustation. Gustation is another chemical sense in that we are again detecting chemicals from our environment. When we're doing gustation, the process of tasting, now we call those chemicals tastants. It's the same kinds of chemicals that we were smelling, but when we detect them with taste, we call them tastants. Just like we had to dissolve odorants in our mucus in the nose to detect them, we must dissolve tastants in the saliva on our tongue to be able to detect them. So once you've written that word saliva, underline, highlight, star it. We have to dissolve all of our tastants for our cells to be able to detect them. The kind of cells that we use to detect gustatory sensations or chemicals that we're tasting are called gustatory receptor cells, just like we had olfactory receptor cells. Gustatory receptor cells are found inside little things on your tongue called taste buds. Now, a common misconception is that when you look at your tongue and you see bumps, you're seeing taste buds. Actually, taste buds are much smaller than those bumps that you're seeing on your tongue. We will talk about those bumps. Those bumps are called papillae. But for now, know that the thing that I use to detect taste sensations are the taste buds, little groups of cells that I find throughout the surface of the tongue. The part of a gustatory receptor cell that actually detects chemicals, detects the tastings, are these little parts that stick up here at the top. These parts are specialized microvilli. Sometimes they're called gustatory hairs. So these hairs are covered in two types of receptors. Some of the receptors are ionotropic and some of them are metabotropic. We mentioned this terminology as it related to smell, in that smell is a metabotropic receptor mediated sense. We actually use both kinds of receptors in taste, and I'll show you what those look like side by side. In addition to having our gustatory receptor cells inside our taste buds, we also have basal cells in taste buds as well. And the basal cells in taste buds do the same thing that the basal cells in the olfactory epithelium did. They make us new receptor cells. We can all be very happy that we replace all of our receptor cells about every 10 days. This is why when you drink that coffee that's a little too hot and it burns your tongue and you can't taste, it's not like you can't taste forever. It's just that you can't taste for a little while. We always can replace those receptor cells. When we look at our tongue, we see some bumps on it. Those bumps are things that we call papillae. So there are several different kinds of papillae, or several different kinds of bumps that are found on the tongue. These bumps look a little different. They're found in slightly different places, and they have different numbers of taste buds on them. So let's talk about these papillae one by one. The first kind of papillae are called valate papillae. Valate papillae are the largest papillae that we have on the tongue. They're found in the back of the tongue, making a letter V. That's how I like to remember these guys. The valate papillae are the V on the back side of the tongue. Now, I mentioned that they are the largest. Because they are so large, they have space for somewhere between 50 to 100 taste buds each. So if we look at our picture here, we can see those valate papillae on the back side of the tongue. If we zoom in on one of the valate papillae, you can see all these little dots here on the side, all these little dots here on the side. Those are all my taste buds. Taste buds filled with the gustatory receptor cells. We do a lot of taste detection here in the very back of the tongue with these valate papillae, making this letter V. The most common kind of papillae, what you would see if you looked at your tongue in the mirror, are what are called fungiform papillae. And fungiform papillae got their name because they look kind of like mushrooms. So fungus and fungiform, that's these fungiform papillae. So fungiform papillae are found almost everywhere on the tongue. They're a whole lot smaller than the valate papillae, which is why they have many fewer taste buds, only 3 to 5 taste buds per fungiform papilla. So all of the little bumps that we see scattered across the tongue, these are the fungiform papillae with much fewer number of taste buds in those. One kind of papillae are only seen in children, and these are called foliate papillae. What we need to know about foliate papillae is that these are located on the sides of the tongue, but again, they're only seen in children, so everybody grows out of these as we get older. I like to joke that this is the reason why children don't like vegetables, because they literally have different taste buds than adults do, and their taste buds do not like vegetables. So foliate papillae, the bumps on the sides of the tongue, they do also have some taste buds in them, like our other papillae, but again, they're only seen in children. The last kind of papillae are called filiform papillae. Once you've written the word no for having no taste buds, I need you to underline, highlight, star this whole statement right here, that they have no taste buds. You should never, ever tell me in any circumstance that there are taste buds in filiform papillae. These do not help with tasting. What they do help with is things like texture. Mechanoreceptors are really good at detecting texture. Or if something is really hot or really spicy or really cold. Remember, those nociceptors could be activated. So filiform papillae do not help me with the sense of taste, but they are present on the tongue. They do help food to move around on the tongue, and we do also collect some information about your food, like its texture or its temperature, using the filiform papillae. As you're studying this material, make sure we know which types of papillae do have taste buds, which types of papillae don't have taste buds. That's my wink, wink, nudge, nudge. I'm promising you're going to see a question like that. When we talk about our taste buds, that remember are in the big valate papillae, that are in the fungiform papillae, that are in the foliate papillae, there are 5 different taste sensations that those taste buds can detect. And everything you eat is some mix of all of these sensations. So let's go through them one by one. The first taste sensation is sweet. So sweet sensations are caused by organic substances. And here's a reminder for you that organic in science means that it's something that has carbon in it. So sweet sensations are what we see in sugars, natural sugars or otherwise. This is the same sensation that we detect as well with artificial sweeteners like sucralose or aspartame. Sweet sensations are something that we would say we have a high threshold for, meaning we need a lot of sugar, a lot of sucralose for our body to register the sweet sensation. That's going to be different when we start looking at some of our other sensations. Our next sensation is the sour sensation. The sour sensation is caused by acids. So acids like citric acid or acetic acid, that's a fancy name for vinegar. Any acids are going to register as a sour sensation on your tongue. We also have salty sensations. Unsurprisingly, salty sensations are caused by salts. So the most common one we think of would be sodium chloride, but it could also be calcium chloride, potassium chloride. When we talk about salty sensations, this is another sensation where we have a high threshold, meaning there's got to be a lot present or I'm not going to taste it. Now let's talk about the sensation that has a very low threshold. These are bitter taste sensations. Bitter taste sensations are caused by a variety of different chemicals. One chemical, Dr. Ahles' favorite, caffeine, in my morning coffee, but also things like nicotine, cocaine or LSD, and also poisons. Let's underline, highlight, star, poisons. See, we have a very, very low threshold for detecting bitter things, meaning we don't need very much present for us to detect it. This is an evolutionary adaptation for us. If something has poison in it, we don't want to have a whole lot of poison before we taste it. That could be deadly. So we can detect bitter very well. We have a low threshold for detecting bitter tastings. We can detect those very easily. The final taste sensation is a fun taste sensation. We don't actually have an English word for it. The closest to it would be savory or delicious. But the word that we actually use for this sensation is umami. So umami is the taste sensation that's triggered by the amino acids glutamate and aspartate. Glutamate and aspartate are 2 amino acids that are really common in things like steak or aged cheeses, Asian foods, and I also want you to underline, highlight, star MSG. MSG. Because see, when you pick up that bag of chips and that bag of chips says, bet you can't eat just one, they're not lying, because they put some MSG in those chips, and you eat one and you say, ooh, mommy, these are delicious. These are savory. I can't just eat one. I've got to eat more. So, what you're tasting in the foods that are particularly delicious, that's this taste sensation umami. Whether it's from MSG being added or it's something where we naturally see these amino acids. Everything that you taste is a combination of these taste sensations. Your brain will process it to help you figure out exactly what you're tasting. So I mentioned for you that our gustatory receptors come in two varieties, either ionotropic receptors or metabotropic receptors. I do want you to know which taste sensations use which kinds of receptors. So if we have something that's salty or something that's sour, the kinds of receptors that those tastants will activate are ionotropic receptors. Now, what I want you to write underneath ionotropic receptors is a term that we've talked about before. The term that we talked about before was chemically gated ion channel. Chemically gated ion channel. In a chemically gated ion channel, what I'm detecting acts as a chemical key and opens up my channel, allowing ions to rush inside so I can detect the taste sensation. So when we talk about salty or sour, these activate ionotropic receptors, also known as chemically gated ion channels. We detect these sensations very quickly. When we're talking about the sensations of sweet, bitter, or umami, these sensations use metabotropic receptors. Remember that metabotropic receptors are my indirect kind of receptors. So a tastant attaches to my receptor, which activates a protein inside the cell, activates another protein inside the cell. Eventually, we're going to open up an ion channel to help us detect things, but it's going to take a little bit longer and it's going to be more indirect. So, 3 of my tastants kind of indirectly activate my gustatory cells, sweet, bitter, and umami. 2 of them directly activate the cells, salty and sour. Regardless of the type of receptor that we've activated though, all of this sensory information is going to be sent to the insula, that inner lobe in the brain. The insula is where I process all of these sensations, where I can figure out what it is that I'm tasting to get a good idea of how something tastes, whether or not I like it. On that note, we've finished 2 special senses: olfaction, the sense of smell, and gustation, the sense of taste.